Now, contrary to the hyperbole you're hearing from the paid professional whores in the mainstream media, this is a lab-created virus, and it's been released, accidentally or otherwise, for several different reasons. So for education purposes, let's first examine the lab-created aspect.
Deadly strain of human swine flu, never seen before.
This is a virus the world has really never seen before.
A strain of swine flu that it hasn't seen before.
And the virus is one health officials have never seen before.
Are we actually fighting a man-made tragedy? Some are asking.
It is an intriguing question. What if the swine flu virus that has killed 61 people and spread to 30 countries was not something that occurred in nature, but was instead man-made in a laboratory? That is the question that Dr. Adrian Gibbs is raising. He's a respected scientist who was involved in the research that led to the development of Tamiflu. He claims the new swine flu virus might have been accidentally created in a lab by scientists using eggs to grow viruses and make new vaccines.
It might be some simple error that has not been recognized.
The 1918 flu, called the Spanish flu, was extracted from the corpse of a dead Inuit woman who died of the disease in Brevig Mission, Alaska, a small Inuit village, and it was recombined with, combined with other forms of flu to create this particular H1N1 that's been declared a pandemic by the World Health Organization. Contains two forms of human flu, two forms of swine flu, and one form of avian flu.
Now, with a little time and effort, you'll find that Madsen isn't the only chap asserting that this is a Frankenvirus. So I hope you'll take the time out to do that research. In the meantime, why don't we hear from one of the scientists responsible for creating this monstrosity? But before we do, let's take a layman's look at how these viruses operate. This will take a few minutes, but it'll be worth it.
But for our purposes, let's represent a virus by this unpleasant fellow. For all his nastiness, he isn't all that complex. In fact, he only has 8 genes. We, by comparison, have more than 20,000. We're going to look at 2 of these 8 flu genes. One because it's responsible for getting the virus into a cell, and the other for getting it out again. Flu gets into cells with the hemagglutinin gene.
So in order to penetrate cells, you could imagine that the hemagglutinin was sort of like a key.
A key which we'll refer to simply as H. And that key unlocks the cell so that the virus can get inside.
If it gets into your cell, it'll take over the machinery of the cell and start making more copies of itself.
But now the virus has a problem. These copies are stuck to that cell. That's where the other gene comes in. It's called neuraminidase, or N for short.
Neuraminidase is critical for release.
Getting back out.
out.
The virus copies use N to cut themselves free. And now each of them, armed with their own H's and N's, are off to infect more cells. In birds, there are 16 different kinds of H genes and 9 N genes. Every flu is some combination of these. Humans have only caught a few. The Hong Kong flu, which killed nearly 1 million people when it broke out in 1968, gets in with H3 and out with N2, so it's called H3N2. In 1918, the virus which killed 50 million people was H1N1. Now there's the avian flu that's got everyone so worried, and it has a new combination of H's and N's, H5N1. The avian flu's H key, or hemagglutinin gene, can open the lock between birds and people. That is, transmit from birds to humans. But it still cannot open the lock between people, spread from one person to another. Yet. But here's the problem. Once it's in our bodies, the hemagglutinin gene can change. It's as if it can fit the lock, but it can't turn it. Which would be great if it stayed that way. But a virus's genetic recipe constantly changes, or mutates, as scientists like to say. And if just the right changes take place in the hemagglutinin gene, those changes could open the lock. Allowing it to spread between people. But exactly what are those changes?
Right, so here we can see it.
Going back to Taubenberger's recipe for the 1918 virus, Ian Wilson and his colleagues at the Scripps Research Institute found the answer to what the 1918 virus may have needed to spread between humans. They pinpointed 2 changes, or mutations.
Only 2 mutations were sufficient to change the virus hemagglutinin to adapt to human receptors.
So, could those same 2 mutations in the avian flu's hemagglutinin gene allow it to spread between humans also? To find out, they tried those same 2 mutations.
To our surprise, we found that, in fact, we couldn't very easily change it with the mutations that occurred in 1918. So that suggests that it might actually be a little bit more difficult, and it might take a little more time for an H5N1 virus to be able to adapt to human lung cells.
And that's good news. The Scripps team believes the H5N1, or avian flu, doesn't seem to adapt easily so that people can infect other people. Back at the CDC, Tumpy is taking a different approach to find out why these viruses are so deadly. Instead of experimenting with tiny variations in a flu gene, Tumpy is testing entire genes. He's looking at each of the 1918 virus's 8 genes one by one to see which ones caused it to be so lethal and which ones should be the target of new antiviral drugs.
And now, here is the interesting bit.
He started with that H-key gene. He took one from his 1918 virus and put it in an ordinary seasonal flu.
A contemporary influenza strain that doesn't kill, and all of a sudden it was lethal.
All signs seem to point to the H-key, or HA as scientists call it, as being at least partly responsible for lethality.
Well, there's something very intriguing about the HA.
So intriguing that now Tumpy is planning to do something pretty radical. Put the hemagglutinin gene from the 1918 flu virus into the avian flu virus to see if he can create an avian flu virus that can spread from person to person.
You want to combine H1 1918 with H5. What's going on there?
I think it'll be important as a set of experiments to understand how H5N1 works. And by mix and matching it with genes from a virus that actually did that quite well, the 1918 virus, we were hopeful that we can figure that out. Well, I think from a scientific point of view, this is the only way to understand how these pandemic viruses work.
Tumpy is one of the few people in the world who has the clearance to work with a live 1918 flu virus.
So if we can figure out how to slow it down studying this virus as a model virus, then perhaps we'll advance our knowledge on the avian H5N1 virus as well.
So that is the process, and that's the sort of thing that they get up to down at the government-sanctioned laboratories. Now, I don't know how much evidence you need, but I think that's pretty conclusive, don't you?